the final day. You see his face. Uh, if we were to give it on the last day of class,
Yeah.
we cut short in his class. We need that one day Friday.
Yeah.
So it's more or less just taking advantage, it's an extra day to give him.
Okay, but it'll probably take, it won't take more than like an hour.
I would say for most people probably have it done 45 minutes.
All right.
It's not a comprehensive final, it's not a final, it's the last day.
Okay, thank you. I was kind of worried about it, but no, not so much.
You're gonna do awesome on it.
Thank you. You too, Madison. You're so nice.
I'm definitely gonna try and get in the same class as you.
Yeah. Tuesday, Thursday.
Tuesday, Thursday. Alright.
We will get a lot done, I think we'll have a little bit of time Friday to do a small review. And as I was just telling these folks up front, the exam on for next Friday, it's just the last exam. It's not a final, it's not comprehensive. It won't be any more than what any of the other exams have been. In this class, we just have a difficult time on getting everything in, so that just gives us an extra day. Yeah, Jay?
Is it gonna be in this room?
Yes. It's in here at 1 o'clock. Now, I won't be here. There'll be somebody that'll give you the exam. I am in a decanal review committee on the team that I have to meet with the provost and some other people on committees. So I'll be at school, I just won't be in here. And they will give you the exam. And like I said, I don't expect it to take you any more time than any of the others did. It'll be 80 to 100 points, like the others have been. So it's just the last exam. It's not a comprehensive final. UC gets really irate. Provost office, we need to give an exam on that day, except in labs. And they want it to be done on that day, at that time specified, because a lot of people want to cut out early to leave at things, and they want people to do it at that time, so, yeah.
Is that done on the
Pardon me, it's on Unit 5, correct? Only Unit 5,
If we have like the group study actually do those two on Friday,
yes. Any of the study things, turn it in with the exam on Friday. You only need one per group, and you need to total how many hours each individual spent. In other words, you should have it listed, these are the times and hours, and then at the top, put a total for each person in the group.
If we all have the same total, is it okay just to put a lump sum?
Yeah, I mean, that's, the way that I do these, folks, is at the end, I take all the points from the 5 exams. I will calculate how many points do you have. Then I will calculate how many points that was possible. I will usually take and do a curve at the end, like, let's say, out of 5 exams, there was 500 points possible. I may knock it down to 475. So if you had 440 points out of 500, it would be 440 out of 475. Okay? And your extra points, your study group points, is just added onto your total. That's not, I don't increase the number of points possible. So let's say I calculated, and on your raw scores, let's say you had 440 points, okay, out of 500. If I knock it down, let's say to 480, then that'd be 440 out of 480. And if you had 20 study group points, that would be 460 out of 480. So none of that changed. It's really helpful to you, yeah.
On the exams where we had a curve, if we had over that curve, do those still
That
count?
still counts as that's extra points for you if you had over the curve, yes. You don't lose anything in here. Yeah? No. At the final grade, whenever I turn it in to the registrar's office, you should get an email saying go to One Stop. Everything will be on One Stop.
What do you mean go on one stop?
One stop is where you can go see all your grades.
Oh, okay. You mean like a
You go-
website or?
Yes.
Yeah,
You
it's part
see
of
on
the
site.
college, it's, if you go there, you've got an account with your login name and everything, and you go into it, it'll show you what your grades are.
Oh,
See right here.
If we think that there's something wrong with the grade, do we email you or?
Yeah.
Okay.
If you got a question about it, if you think something's wrong, you just contact me. You'll have to do it by email because, school will be out, but no problem. I try to take a copy of everything with me home, so I've got it there. See? Any other questions? That's pretty much it.
So we have to turn in our hours with our exam on Friday. So how should we do that? Like paper clip it or something? Okay.
Just put, there'll just be a stack and just pop it on there.
Okay.
You can, whenever you first come in. Now, let's not, by the way, excuse me, since I won't be here, make sure Nick, the lab guy, is the one that's gonna be in here. So just tell Nick that you also turn in your study group points, just put them in stack, put them with papers, okay? It will go smooth, folks. Now, last time, if you recall, one of the big questions, or the big areas, and I'll go over total review on all of it on Friday. But remember, transcription and translation, certain things to remember about it. Transcription is copying a gene in the nucleus. That copy is called messenger RNA. It leaves the nucleus and goes to the ribosome. It is at the ribosome where the protein will be translated or made. Transcription means to write the copy of the gene. Translation means to make the protein. We looked and you saw that ribosomal RNA is actually the ribosome. Remember, there are two kinds of ribosomes. Those two types are the free and the attached. If the protein's being made in a free ribosome, that means the cell will use it. Could be made to repair an organelle, or it could be to go in the membrane of the cell, whatever they need the protein for. If it is made on the free ribosomes, on the attached ribosomes, then it will be exported out of the cell. That is the function. Part of the function of the ER and the GOLGI is to prepare that protein to be taken out of the cell. Usually it will be dumped in the blood and taken to different places in the body, similar to insulin, the way insulin occurs.
Where are the attached proteins? Is that on the membrane? Attached to the
Where's the what?
membrane? Attached ribosomes, is that on the membrane? Attached to the
No,
membrane?
the attached ribosomes are on the endoplasmic reticulum.
Okay. On here.
It is called RUF, ER, because it has those ribosomes attached to it.
Oh, yeah. Makes sense.
The ones that are not attached are just called free. They are in the cytoplasm. And we will look at this little picture real quick that we looked at last time, towards the end. This is one of the best setups that, to explain all of that. Remember that the RNA polymerase is the enzyme that is going to be making messenger RNA. And it will separate short spans, and then there will be these nucleotides, which are in the nucleus, will then be put into place, match up, and then we get this strand of messenger RNA made. Remember, polymerase, RNA polymerase. When we go to look in the next section on how DNA is copied, the enzyme will be DNA polymerase. So here, RNA polymerase, and remember, these parts that we had, the promoter, and then we've got the actual coding segment. At the end of that transcript is called the polyadenylation signal because it's referring to adenine, a series of adenines. At the end of that signal, it goes from, as you see the AAUAA, just into straight group of adenines, AAAAA, that's called the poly A tail. Before messenger RNA becomes functional, remember, let's see, where's that at? We had to get rid of these introns, remove those. Those are the non-coding parts. They probably play a function in the transcription process. I don't know that anybody knows exactly what that function is, but they do play a role. We may not know what it is. So those introns are removed, and then we get the coding segment with the cap and the tail. That is done, this modification, removing the introns, are done in sections called spliceosomes. Those are little structure that will hold that transcript, remove the segment that is the intron, and then reattach it. Ribozymes. Remember, RNA can also act as an enzyme. Ribozymes are catalytic RNA that function as enzymes and do the splicing of RNA. So remember, RNA is going to be spliced by ribozymes. This little picture right here, remember, I told you, hopefully, we've got one end. This is transfer RNA. One end holds a particular amino acid. The code for that amino acid is the anticodon, the reverse. That reverse codon will then fit where it's supposed to on that transcript, or the protein that's being made, actually, or the RNA. This is the RNA. It just serves as a template. So these come into a specific order. The new amino acid comes in with the tRNA in one spot, that is A. It moves to this spot, which is P, attaches its amino acid to the growing chain, and then exits at E. So it's E, P, A, real easy to remember. It enters at A, donates its amino acid at P, and then exits the ribosome at E. Then we have that growing protein chain. That is what a transfer RNA looks like. You don't have to remember that. I just want you to see it so if you ever hear in the future that you will have an idea of what it is. And this is just the end. This is the same blue thing that we just looked at. These blue transfer RNAs, this is chemically how they're put together. Notice they're put together with nucleotides. They will have bonds that hold the shape and things together. And at one end is where actual amino acid will attach. This is the anacodon side. So this will only attach where there is a U, and a C. Okay? Now, that is pretty much the shape, but remember, whether we are building a messenger RNA or if we are building the protein, there are three phases: initiation, elongation, and termination. Start, build, stop. Another thing is, remember, we're not giving you every single part that's involved in this, such as these factors. There are little factors, proteins, that will attach to these things and initiate them to do things. Here you see we've got a protein that's getting ready to donate its amino acid. That will be done by this. That causes it to release, and then these come out. So there are proteins, whether we're copying or making messenger RNA or the proteins, there are other proteins that help to function to get that going, start. Yeah,
Do we need to remember the release factor?
no, just know that they're there, that there are, I'm not gonna ask you that on the test, but just understand that there are other things involved in here. We're not giving you a 100% complete list of all of the structures and molecules that are involved in this. You're getting an overview. You could have a semester-long course just on these factors and things. There's so many. This picture here is a very good wrap-up overview of transcription and translation. Transcription taking place, copying the DNA, making the pre-mRNA. Once we've got that pre-mRNA and we remove all of those bad parts, the introns that we don't need, we come down, we've got the actual mRNA functional that has its 5' cap, the poly A tail for protection of the transcript. They leave the nucleus through the nuclear pores. They go to the ribosome, whether it's free or attached, doesn't matter. And then we have the transfer RNA that brings in the amino acids and it starts that growing peptide chain. Remember the three kinds of RNA: messenger, which is the gene copy; transfer, which will carry the amino acids; and rRNA. The ribosome itself is called ribosomal RNA. So know what transcription is, what translation is. Transcription's writing the gene copy, translation's making the protein. And what I was referring to, that there's other factors that we don't always talk about, look right here. There is actually an enzyme, aminoacyl tRNA synthase. It's going to be that enzyme that's going to attach the amino acid to the proper tRNA. It will not do it with any tRNA. It has to be the proper one with the proper anticodons. These things right here, those amino acids and things, they're just out in the cytoplasm of the cell. tRNA will collect the correct ones and then bring it to the ribosome. Now, try to keep straight what we have just talked about. We have talked about how protein is made. We have talked about how messenger RNA, the transcript, is made. Then how it is used with transfer RNA and rRNA to make protein. Now, that part's finished. we're going to look at, when we talked in mitosis, for example, that we said in interphase that the DNA is copied. This next chapter is on how that DNA is copied during interphase. Okay? When we've got one cell that's going to be copied and become two, this next chapter is talking about how the DNA is actually copied. It is not completely different than how messenger RNA is made. Some different components and different parts, but keep in mind, where is this going to be taking place? During interphase, before mitosis, before the actual replication. We said in the cell cycle that we've got three phases. Interphase, where the copying is done. Mitosis, the mitotic phase, where the chromosomes are now duplicated and they start to split apart. Before we can make chromosomes out of them, we have to make DNA. So that's what this chapter is about. Now this is called the molecular part of inheritance. Remember, when we talk about heredity, we're talking about DNA passing down genes. Well, this is how those genes are copied, how some will go into one cell we looked at through meiosis. This is how they're copied and transferred. This is history. Just remember, we really didn't know too much about DNA, that its existence till Watson Crick came along in the 30s and came up with the idea of that spherical helicase of DNA. Some of this you already know. If you remember back, we said there are two kinds of nucleotides, purines and pyrimidines. The difference is purines have a double carbon ring. Pyrimidines have a single. Now, which one of these are purines? Here is a double ring. There is a double ring. So adenine and guanine are purines. Thymine, cytosine are pyrimidines. Remember, when we were talking about making RNA, RNA uses uracil instead of thymine. They code for the same. If we look at DNA and we talk about the spiral helix, you've got the vertical parts and you've got the rungs, like of a ladder. This vertical part, every one of these nucleotides are made up of a nitrogen base attached to a sugar, a ribose sugar. In RNA, it's just ribonucleotide or ribose. In DNA, there's oxygen removed. It's called deoxy. So we've got a nitrogen base, the ribose sugar attached to a phosphate. It is the phosphate and the sugar that makes those vertical parts of the ladder, the sides. Phosphates attached to that sugar. That phosphate attaches to that sugar, so on, and we build the ladder. In the middle is the coding part. This holds the structure of the molecule in place. This is the actual code. The code is what order that these or nitrogen bases are placed. And that's what we will look at. So this shows DNA. Notice when they show a purine, it is longer than the pyrimidines. These little dots represent the bond. The bonds, remember, are hydrogen bonds. Why do we want them to be hydrogen bonds?
So that we can separate them when DNA is
They
replicating?
are not a terribly strong covalent bond that's hard to separate. Hydrogen bonds will separate easily, so when enzymes come along and have to unzip DNA to copy a gene to make RNA or whatever, they can be easily broken apart. Also noticed, this is the 5' end over here. Here's the 5' end. That is called anti-parallel, which one goes in one direction and the other one goes the other. But because of that, when you put their codes together, they will code for the same amino acid on each strand. And that is showing you just what, if you had these in the shape of molecules, that DNA would look like. So remember, why do we have a purine and a pyrimidine? If we had two purines, they would be so large, there would not be enough space to fit them within that structure to make the bond. If we had two pyrimidines, it would be too narrow to make the bonds. But when we have a purine with a pyrimidine, it's just right. It's like Goldilocks of the three bears, too hot, too cold, and just right. Purine and pyrimidine, because of the space, is just the right amount. And these are called the complementary base pairs. They complement each other. Adenine and thymine, guanine and cytosine. The information they will code for will be the same, but they are different structures, and they are designed like a lock and a key, one to complement the other. Note also, as, yeah,
Are those the two pairs that always bind to each other?
yes, it's always going to be adenine, thymine, nothing else. If you've got thymine on one, it'll be an adenine below it. If you got an adenine on one, it'll be thymine, always those pairs, yeah.
Could you go back to the last slide? I actually have a question about when it's showing the purine and the pyrimidine, actually, like the size. So why is there only one hexagon on this side, on the right side?
Was there only one?
Oh, sorry. So for the purine and the pyrimidine together, why is there only one, what is it, pyridine on the right side?
Because it's this space. This space is predetermined by the phosphates and the other adenine or the bases. So you got a specific space. Only a purine and pyrimidine can fit in that space and still have room to make the hydrogen bond. If you're too far apart, the hydrogen bond won't form. If you're too big, when the hydrogen bond forms, then they won't fit within the ladder.
I see. I was mistaking the on the left, yes, as two different ones, but that's actually one purine. I see. Okay.
One purine, and that's a purine and one pyrimidine.
Okay. Thank you.
There's going to be other things, once again, happening here that we don't talk about, other little structures. You'll see them as they just say transcription factors. They don't tell you what they are, but they're going to be molecules that are helping things do it. What we're trying to do is give you that basic overview. You should remember the two guys that designed or come up with the concept of DNA is watching the Crick. And there's a little story about that. I've done do a lot of work, build things and so forth. One of the things I was working on was making recurve bows. Well, to make recurve bows, you need special wood, Osage orange. A buddy of mine, actually a guy that I had in school, became principal and so forth later. They have farms down in Ripley. Now, if you go up on high hills right over the river down there, he knew one guy who had a farm down there, and they were clearing it off like they had osage trees off. So we went down there to find some logs where they could take them out. One of the guys was an older fella, and this this has been 15 years ago. Big guy come up, bigger than me, wearing overalls, your typical farmer that you would expect in this area, but he wasn't. He had been a teacher, was a graduate of Harvard, and he actually, in his class, he was in one of the small classes at Harvard when Watson and Crick came in to tell that they had just discovered DNA in the 30s. So this guy was obviously much older fella. But you'll be surprised, don't ever assume about people you run into, because they fool you a lot. And this is not the guy that you would think that ever even been out of high school, let alone graduated from Harvard and listened to Watson and Crick. That's a big deal, folks, to say, I was there whenever they made the announcement in my class that they found that. So remember Watson and Crick. They were the two researchers who came up with the idea that there is a molecule called DNA, and they came up and made that double helix type model. When you see that term, that the two strands of DNA are complementary, think of it this way. They both strands code for the same amino acid sequence. They're just using different directions, basically. Notice it says in DNA replication, the parent molecule, the main DNA molecule, unwinds. Now, whenever they say it unwinds, it just doesn't decide to do it. There are enzymes called helicases that actually make them unwind. And then two new daughter strands are built based on each one. You've got one strand with a code, so you put the anticode down here on that one, and this one's got the code, you put the other code on that one, and you wind up with two strands. Here's an example. This is our main strand of DNA. It's going to be separated. That's going to be separated by an enzyme, helicase. And then other enzymes are going to say, okay, we've got a guanine right here, so we need to get a cytosine and match up to it. Then we've got adenine, thymine, and so forth. And they will build, this is called the anticodon, or the other strand, to match that one, to complement that one. This strand that was separated, same thing happens to it. So if you notice, this strand is the same code as that one. And this strand is the same code as that one. So they just copy. That is done by enzymes. Now that, when we say in interphase, that DNA is being copied, this is what's happening. Later, after the DNA is copied, then chromatin will come into the picture, and they will turn into chromosomes, and then get separated into two different cells. After they get into two different cells, then that chromatin wrapping protection will go away. So now we've got the bare strands of DNA that are functional again and ready to be separated to make copies of messenger RNA and to make a gene, or a gene, make proteins. The copying of DNA is fast. And notice here, you don't have to remember all of these enzymes, but a dozen enzymes and other proteins are involved. Where this begins is called origins of replication. I think Katie had asked the other day that, like RNA, does it have to start in one spot, and there was the promoter. Not the case. It's the case here, but there's an exception. Here, one of the reasons that it's not just happening in one place, it can happen in a bunch of different places, then it all joins together. That we'll get to see. And notice here it says, in a eukaryote, us, there could be hundreds or even thousands of places where the replication starts. And that's why it can happen so quickly. Notice it says it goes in both directions until that entire DNA molecule is recreated. Remember now, we're not just talking about copying a gene, we're talking whole strand. Everything on the DNA is to be copied. These show some of these replication. They're called replication bubbles. Notice they go in both directions up here. They're not just copying in one direction. This one will start to go that way, this will go, eventually these will meet. We've got the copies going. These little light blue represents the copies being made, the dark blue represents the parent strands. This shows that being occurring, these bubbles and things in through here. Now, so remember, where replication starts is called the replication bubble. That is where it's going to be unwound. At the end of each replication bubble is a replication fork Y. Right there, you can see replication fork. Shows you right there. That's the Y-shaped region where it starts to elongate. Helicases are the enzymes that unwind the DNA molecule, the double helix. Single-strand binding protein bind to and stabilize this single-stranded DNA. So what they're saying is whenever you got the DNA's bound together and now it's open, there are other proteins that come in to stabilize it so it's not moving and wobbling around. It's got a stable base. Topoisomerase. What kind of molecule do you think topoisomerase would be? What group? Favorite enzyme. You can tell because it ends in ASE. This is one that is there so that the little helicases can't get too overexcited and unwind too much. They put a limit on how much it can be unwound. It corrects overwinding. So here is the topoisomerase, that big enzyme that's just there to make sure everything doesn't get out of hand and stays in place. Here is the helicase, who's just like unzipping a zipper. You've got these hydrogen bonds that hold the bases together. It breaks those hydrogen bonds, moves this way. The isomerase will give it some leeway and then move down, but not let it hold too much. These, notice, are these single-stranded binding proteins that are there to stabilize the DNA. And we will see what this is here, but notice there is one little section. The copying of DNA must include RNA. It is a small section called RNA primer. That is going to give a place for the big enzymes that's actually going to put the new nucleotides in place. It gives it a starting point. And this, as you can see, is another enzyme, enzynase, called primase. It is going to cause that RNA primer to be attached as a starting point. And as we said before, RNA polymerase builds RNA, DNA polymerase will build DNA. That initial nucleotide strand is called a short RNA primer. The enzyme primase starts an RNA chain, adds RNA nucleotides one at a time using the parental DNA. This is going to be the starting spot. And notice that primer, the RNA primer, is not going to be very big. It says 5 to 10 nucleotides long. The polymerases elongate DNA. They cause the DNA to grow. They add the proper nucleotides in the proper place. And note,
I really need to just keep going.
there are some folks in the government whose job is to sit and say, what is the most annoying sound that we can come up with to aggravate and interrupt people? And you heard what they come up with, and they probably make big money. Notice, DNA polymerases elongate DNA. They require their primer, which is RNA, and they have to have that strand of DNA to start with. And notice the rate. The rate of elongation in human cells is about 50 nucleotides per second. Now look how fast bacteria can replicate, 500 per second. So bacteria can obviously has a very much quicker reproduction rate than the cells or DNA in people. 50 per second in humans. Each nucleotide that is added to a growing DNA chain is a nucleoside triphosphate. DATP supplies adenine to DNA and is similar to the ATP of energy metabolism. So, there is an actual nucleotide called dinucleotide ATP, and its purpose is just to give energy. You don't have to remember too much, just remember DATP supplies the energy. So this shows how these strands are being made. It comes in as a triphosphate. It's going to lose those phosphates, and then the strands will keep, the bonds will be forming. So basically these nucleotides are carrying their own energy. They start out as a triphosphate and wind up giving off to just to make the nucleotide. DNA polymerases, do remember this. They add the nucleotides to the 3' end of the 3' end. Therefore, a new strand can elongate only in the 5 to 3' end. In other words, they're adding them on the 3' end. If the 5' is on the left, 3' is on the right, they are going to be added to the right. It will be the 3' end. Along one template strand of the DNA polymerase synthesizes a leading strand continuously moving towards the replication fork. There will be two strands here called leading and lagging. So this is giving you the definition of the leading. It moves towards the replication fork. So here's the leading strand. Now, whenever they're saying the leading strand and lagging strand, they're not talking about the two main strands of DNA. It's what's being made, the copy. Notice, here's the replication fork. So one will go towards that replication fork is called the leading strand. This one will be behind it and is called the lagging strand. The lagging strand will be in little pieces. It will not be in the one big continuous piece that you see. Same thing for the top. Lagging and this copying in that direction. Remember, we're copying 5' to the 3' direction. But same thing is happening on both. It's just the directions. To elongate elongate the other new strand, called the lagging strand, DNA polymerase must work in the direction away from the replication fork. Notice this is moving away from the replication fork on this end. We have the midpoint marked. From the midpoint towards the fork is called leading strand. From this fork going away from that fork is called the lagging strand. You're not going to have to identify the leading and lagging. Just know what the definitions are. And here's why we want you to remember that. That lagging strand, notice it's in segments. It's not a continuous piece such as this, like leading strand. The lagging strand is synthesized as a series of segments called Okazaki fragments, which are joined together by DNA ligase. I'm assuming that the Okazaki was the name of the guy that discovered these fragments. These will be joined together by an enzyme called ligase. And this just gives you a little rundown how they work. These are the things we've talked about. Now, you're going to see that there are 3 different kinds of DNA polymerase, 1, 2, and 3. I'm not going to ask you which one is where. It will tell you a little bit later here. But this just shows you how these parts are tied together. And this is to remember. This is, as it says, a complex. It's a, it is just not 1 or 2 little things happening here. There are a lot of these proteins, a lot of things involved. There's an overview of these strands and what's happening. Now, we will stop there. That gives us 15 or 16 slides for next time. And we will talk about how DNA proofreads its work to make sure that it puts out the proper product. Any of you want to turn in your notebooks, you can put them right up here.